And then and our next speaker is Lotfi Merabet. Lotfi did a very brave shift to the emerging new field of cortical visual impairment a number of years ago, and now he's really leading very much the efforts in the field and doing really fantastic work. So, Lotfi.
Thank you. All right, good afternoon, everyone. I remember about 15 years ago, I came to present at Smith-Kettlewell. Despite that, I was still invited back. So I take that as a good sign. Thank you, I appreciate it. Again, as always, for our friends who are visually impaired, if I don't describe any of the figures or anything well enough, please just raise your hand. I'm happy to spend more time. I'll do my best to further give you details. I want to start this presentation off. Forward. There we go. A little bit from a public health perspective, because in many ways, this wasn't a project that we looked for. This was a project that found us. So what I'm showing you here on the left is the Perkins School for the Blind. It's a little bit outside of Boston, in Watertown, Massachusetts. As many of you know, it's the first and oldest school for the blind in the United States. It's a school with tremendous history. Helen Keller was a student here. I can also tell you that when I was an optometry student, this is where I interned. So it had an enormous effect in terms of my ideas and what I wanted to do in terms of my research. As many of you know, and as Laura mentioned, I started off in profound ocular blindness, and then I would say about 5-6 years ago, made the shift towards brain-based visual impairment. Now, this is how the story comes. If you looked at about 50-60 years ago, the sort of the classic Perkins kid, you would find a child who was profoundly blind due to ocular causes. This could have been cataracts, this could have been, for example, retinal issues, retinopathy of prematurity. And about 20-30 years ago, something happened. The profile of children enrolled at Perkins changed dramatically. We are now seeing children enrolled at the school who are not necessarily profoundly blind, at some level of visual function, but we're presenting in a much more complex fashion with other issues: hearing, motor, cognitive, language issues. And what was challenging is that these kids were not responding to that same classic, traditional, tried and true curriculum that was being taught at Perkins. So teachers reached out to me, parents were reaching out to me and saying, what's going on here? How can you have two kids with, say, 20/20 visual acuity each, one brain-based and the other ocular-based? Why are they so different? And that was very, very much the impetus of the study. What kind of problem, or what's the magnitude of this problem? Here is just a pie graph showing you roughly about 200 kids enrolled at Perkins. Over half of the children have a diagnosis or suspect a diagnosis of cerebral visual impairment, or CVI, this condition I'm talking about. At the same time, the National Eye Institute very recently published as part of their strategic plan that CVI is really considered an area of priority that we should think about. In terms of the epidemiology, most of this work comes out of the UK. This was work by Kathy Williams, and she estimates that in school children age, at the age of school children, I should say, that's roughly 1 in 30 children. That's one child in every classroom that potentially has CVI. So a little bit more in terms of definition. Cerebral visual impairment or cortical visual impairment has a functional definition that was developed by Saki and colleagues in London. And they said that CVI is verifiable visual dysfunction, which cannot be attributed to disorders of the anterior visual pathways or potentially co-occurring ocular impairment. So it's a very broad general definition that really highlights the brain-based nature of these visual impairments. Now, that's not to say that these children don't have eye issues. Many have strabismus, many have ocular nerve head issues. But the key issue here is that their visual impairment is not explained by what's going on at the level of the eye, or solely what's going on at the level of the eye. It's a brain-based issue. The presentation is very, very heterogeneous. Some have normal visual acuity, others have profound blindness, but the majority of these kids have some level of visual function. Many have visual field impairments, contrast sensitivity impairments, and also ocular and motor issues. But it's important to note that visual field deficits and impaired visual acuity are not sine qua non for the definition or a diagnosis of CVI. In fact, many of the children that we work with have good visual acuity, intact visual fields. As you might imagine, it's difficult for these kids to register. For example, they don't qualify for the statutory definitions of visual impairment. So these kids fall through the cracks. Last thing I'll mention, which is absolutely important, is that often these children have a medical history that is consistent with some sort of perinatal or early neurological injury. So something like a hypoxic or ischemic event. There are also many genetic and metabolic disorders like Rett syndrome, in fact, that are associated with this condition. Neurological comorbidities, like cerebral palsy or epilepsy. And often there are neurological findings associated with this as well. So periventricular leukomalacia, which is often associated with cerebral palsy, for example, and I'll show you a little bit more about that. But again, damage revealed by structural imaging like MRI is not a requirement for a diagnosis of CVI. There are many kids who have CVI who don't necessarily have any obvious structural issues. Sure, no problem. Do you want me to make it higher? Yeah. How's that? Is that better? No? You. Is that good? Yeah. Better? Yeah. Better? Excellent. Thank you. So this was a study by Lisa Fazzi and colleagues from Brescia, Italy. These are collaborators for us. And they did really a seminal paper that was published in 2007, where they looked at over 121 patients with cerebral visual impairment or visual impairment associated with neurological disorder. Roughly half of them were born premature and the other half were born term. And you can see on the right here that the causes of CVI were quite diverse. The majority had some sort of cerebral vascular accident, but there were many other causes as well, like metabolic disorders, genetic disorders, and so on. They then did a systematic review of what type of clinical profile that these kids had. And many of them had refractive errors, they had impaired visual acuities, as you might suspect, impaired contrast sensitivity, and going down the list. And what was perhaps the most striking issue is the most common deficit was related to impaired visual perceptual abilities. That was the most classic finding that they found in this particular subpopulation. And the way that they did it in this particular study was using a neuropsychological test called the developmental test for visual perception. Now, I think that neuropsychological testing certainly has its place, but there are limitations with this technique. First of all, they were never designed for kids with CVI. And second of all, if a child does this pencil and paper test, there are many reasons why they could have done poorly. Was it a visual acuity issue? Was it cognitive issue? Was it somehow perceptual? Was it a hand motor issue? And as was raised earlier this morning, I certainly think we can do better. And I'll show you a little bit of things that we're doing in this area as well. Last thing I'll mention before going into the actual data is it's useful to separate the visual perceptual issues that we see or the visual impairment issues in this population at two levels. What we call lower order issues. So these are visual, ocular motor issues. And at the same time, these higher order issues, which are more related to perception and so on. This was a framework that was proposed by Xander Zudek in the Netherlands. So CVI, these higher order issues, this is what the lab largely spends its time working on, is often there is the presence of these unique higher order visual deficits related to visual spatial processing, things like complex motion, visual search, and awareness of their surroundings. And Gordon Dutton has coined the term dorsal stream dysfunction. In other words, that somehow associated with the dorsal stream, visual spatial processing issues and so on are perhaps very more prevalent than what we see in terms of impairments of the ventral stream. So these are the type of things that kids will tell you. They'll say, I have a hard time walking, in busy crowds, for example. I hate really cluttered classrooms. If you show them their favorite toy, they'll look at it and say, yeah, that's my favorite toy. But if I put it in a toy box, they can't find that toy anymore. And what they do is we call them dumpers. They take their toy box and they dump it out and they have to spread them all out in order to find their favorite toy. Or they may have a hard time finding a sibling or a family member in a crowd. This is not visual acuity. This is clearly an issue of their ability to process the complex visual world. So how did we get at that? So we developed virtual reality-based tasks that were ecologically valid and had high behavioral relevance, combined with eye tracking to get a sense of how these kids were processing the visual world around them. Because where you look, where you don't look, tells me something about how you're interacting with the visual world. So the first task we developed is called the virtual toy box. On the left, you see a control, and on the right is a CVI who's age matched. The task is very simple. There's a blue truck that's placed in this 5x5 array. And we change the number of unique identifiers, and all the child has to do is just simply make a saccade to where the blue truck is located. And I want you to concentrate on that yellow bob. That's the actual recording. And what you find that while the control makes a very simple ballistic movement, look how much more complex the pattern is for the CVI individual. Much more circuitous, much more complex path to get there. They get there, but much more complicated. Here's a color match. Here's a toy that's the same color. You see how the CVI individual has to check both before they actually decide? And here we make the environment really complicated, increase the clutter of the visual environment. And you can see in this particular case, the CVI child missed it completely. Again, this is not visual acuity. There's much more going to it. So I know what you're thinking. You're saying, well, this must be somehow some underlying ocular motor issue. Well, that's not actually the case. Here are the same two individuals where all they have to do is simply make a saccade. Hopefully I'll get back to it. Here we go. They have to make a saccade to the blue truck by itself. And you notice how, again, both the CVI and CVI participant are able to make that very robust saccade to it. So again, this is a function of the complexity of the individual environment, not necessarily a baseline oculomotor issue. So here's the task again, the virtual toy box. What we do is we have the child pick their toy. It could be a blue truck, an orange basketball, or a yellow duck, because we want them to identify the toy in isolation, and at the same time has much more of an immersive feel of the task as well. And there are 3 task levels: low, medium, and high. It's a 5x5 array of toys. The only thing that changes is the number of unique toys in the array. So our thought is, as the toy number, the unique distractor numbers increases, the task is going to be harder. And this is what we find in terms of the responses. So in blue are the responses, success rate in controls, and in orange is the success rate for CVI participants. And the first thing you notice is overall their mean is lower, so they have a lower success rate than they do in controls. But look at the variability that you see in the CVI group. It's absolutely tremendous. Some do very, very poorly. Many are clustered in the middle. But there are some who do very, very well. In fact, our best performing individuals with CVI do better than our worst performing controls. And it's that overlap that really makes the diagnosis very, very difficult. At the same time, if you plot these same individuals, you separate the CVI group by those kids who have 20/20 visual acuity versus those who have 20/30 or worse, they actually look very, very similar. So high visual acuity or good visual acuity does not make this problem go away. This is clearly an issue in terms of their processing abilities. Finally, if you plot reaction time performance as a function of task difficulty, so the y-axis is reaction time, the higher you are, the slower you are. Moving to the on the x-axis to the right is a question of task difficulty. What we find in controls is that we have this very, very flat relationship, right? So you can see that the reaction time is virtually the same, no matter how hard the task gets. Here it is in CVI. We have this load effect, that as the task gets harder, the reaction time gets longer. So they're very, very sensitive to the complexity of the visual task as well. We've combined this task with EEG. So this is a nice system because it's wireless. So it's recording EEG signals as a function of doing the oculomotor task in terms of the toy box. It also has a motion detector. So it records movement in 3 planes, and we can actually progress the head motion out of the pre-processing signal as well. I'm showing you here visual evoked potential reportings from the occipital cortex. In blue, you see this nice deflection here in controls. In orange, what's interesting is the CVI group, how these early potentials seem to be largely intact in terms of amplitude, but it's this late potentials, P300, for example, that seems to be much more blunted and reduced. And that's interesting because we know that these later potentials are related to top-down processing, again, related to these perceptual issues. We took it a step further in terms of the analysis, and what we've done here is time frequency decomposition. So what I'm showing you here, again, on the y-axis are various frequency bands, and on the x-axis is the time after stimulus onset, which is the dashed line. If you look here, there is this very, very nice, really conspicuous blue bands, and this is referred to as alpha desynchronization. Those of you who are familiar with EEG recordings know that this is a very robust signal that comes and can be recorded from occipital visual cortex, which is related to visual perceptual functions. Here it is in CVI. That alpha desynchronization band is virtually absent. And on top of that, the other frequency bands, namely theta and gamma, are almost seem to be dephased. And that's interesting because we know that alpha synchronization, or one of the ideas of alpha desynchronization, is that it's like a clocking mechanism. And we think that this somehow relates to the fact that there is often response latency in these kids as well, so that the synchronization of visual signals in their brain seems to be somehow impaired. And this may be a neurophysiological marker for that. The second task we developed is called the virtual hallway, because we also know that these kids struggle in terms of finding individuals in complex environments. So we created this virtual hallway, I should say, and the task is quite simple. You have to choose a principal of fictitious school, and which are gender and age, or race matched, I should say, and then we put them in this hallway where the crowd increases in terms of the population size. So low, medium to high is the number of individuals in the crowd. I'll show you a video as well. Left is in the control, right is the individual with CVI. And you can see how the control participant locks on to the principal and then follows them off the screen, whereas the CVI participant still searches, gets there, but you can see that there again, there's that response lag in terms of their response as well. Here again, very similar to the story that we saw in the case of the toy box. Overall success rate is much lower in CVI than it is in controls, but again, there's that large variability. I'm also showing you here what's called the off-screen percentage. That's how much time that the individual leaves the screen, stays on task. And you can see it's very similar in CVI and control. So this isn't about CVI kids being off screen more often. They're actually very, very similar. They're on task. So again, related to these processing issues. Very similar to what I showed you in the toy box. There's this clear load effect in the case of CVI that we don't see in the case of controls. And a third population that we started working with are children with ADHD, attention deficit hyperactivity disorder. So to get at this sort of idea that, okay, here's an individual who has attentional deficits, but does not have this early neurological injury. And here's what their profile looks like. So again, very, very similar in terms of that load effect, but overall their baseline is much, much lower than what we see in CVI. So CVI, there's much more going on than just attention deficits. There's clearly processing issues as well. We've recently combined our task, the virtual hallway, with fMRI. So here what you see is the same sort of load effect from a behavioral standpoint. That's 5 minutes, that's perfect. So what you see is that same load effect that we saw with the toy box. And here are now the responses that you see from fMRI. And left are the controls, and right the CVI. Overall, nice responses in occipital areas, parietal areas, and temporal areas. And we did a region of interest analysis, looking step by step in various areas and comparing the response profile to task demands in controls and CVI. What you see in early visual areas, V1, V2, and V3, the profiles are quite similar. So they increase in bold responses as a function of task demands, but they overlap. So what that tells us is these behavioral changes or differences are not attributable to what's going on in early visual cortex. It's got to be somewhere else. So marching down or over dorsal stream, this is what we see. So along dorsal areas, you notice that the response profile is much lower in CVI than it is in controls. And this would be indeed consistent with dorsal stream dysfunction, right? So again, this is sort of neurophysiological correlate to that. What about ventral areas? So we did exactly the same thing. And what was interesting there is we saw the opposite profile. When you look in ventral areas, now CVI is responding stronger than in controls. So what is going on here? In the context of dorsal stream dysfunction, could there also be ventral stream compensation? There is an alternative interpretation to this, is that what it could also represent is impaired suppression mechanisms as well. There's a lot of fMRI evidence that suggests that when there's a strong visuospatial task, often what ventral cortex is shut down in order to sort of route resources to dorsal areas. So maybe what we're seeing is in fact dorsal stream dysfunction in the context of impaired suppression mechanisms as well. Next thing I'm going to share with you is we're now getting into wearable eye tracking technology. This happens to be the Tobii Pro device. That beep or that bing that you heard there is the calibration. And here is just me looking in the lab right now. Here you can see me reading the letters off an eye chart. So at distance, it works really quite well. You can see the various saccades. Here I am looking at a near point chart here, or not a near point chart, I should say a near task. This is me jaywalking in Boston. And you can see that when you're outside and walking, how things are much more dynamic in terms of the recording. And here's a visual haptic task that we started doing as well. So what you see here is as the circle gets bigger, that is more time being spent on a particular fixation point or fixation area. And here I'm just showing you the software analysis, shows you the registration of how the software is able to register on top of a particular video based on a static image, what the gaze pattern looks like. So once you have this, you can generate heat maps, you can generate sequences of gaze points as well. And again, the larger the circle, the longer the child fixates, and you can see where they've looked and where they didn't look. So armed with this, you can go back into all these real world tasks. You can see how a child interacts in a classroom through the hallway, for example, or finding their favorite toy. And recently we've been going back to this idea of neuropsych testing. So what I'm showing you here is an individual with CVI doing that developmental test for visual perception as I showed you. So the large image here is this individual with CVI, and on the right, what I'm showing you is the accumulation of fixation points as they do the developmental test for visual perception over time. So he's doing the task here. Again, those are all his fixation points. And what happens here is if you compare a control participant, so the top row and the third row is the target, the second and the fourth row is their actual reconstruction, you can see that as the stimulus or the target gets more complex, there's more and more fixations. There's progressively more fixations. Here it is in CVI. So on the net, many, many more fixations. You'll also notice that the CVI participant looks back at the target more often as well. And the other thing that's interesting is you see how the control uses their parafoveal vision to actually draw the target, whereas the CVI participant is essentially looking right at the tip of the pen as well. So a very, very different profile. What's interesting in this particular case is I sent these two images to a neuropsychologist, and they said nobody here has CVI. But this child was already diagnosed with CVI. So clearly the eye tracking sees more than what the neuropsychological testing would suggest. Last thing I'm going to mention is we started looking at a diffusion-based imaging as well to try to get an understanding of how the brain is wired differently in the case of CVI as well. So we use Hardy, which is high angular resolution diffusion imaging. Again, on the left is a control, and on the right is an individual with CVI. They're rotating here. I'm showing you the outside cortical surface, and these are the white matter projections inside. We then asked the software to see the occipital pole and to show us what visual projections emanate from the occipital cortex. And you can see that in a control, there's this large pathway going from the occipital pole to parietal cortex to the frontal cortex. This is the superior longitudinal fasciculus. This is the neuroanatomical correlate of the dorsal pathway. In contrast, the occipital pole down the ventral pole, this is the inferior longitudinal fasciculus. This is the neuroanatomical correlate of the ventral pathway. Last slide. And in case of CVI, you can see that those pathways look very, very different. And in particular, the dorsal stream seems to be more impaired than the ventral stream. We're also interested in other pathways. This is the inferior frontal occipital fasciculus. This is interesting because it goes from a parietal occipital through ventral into frontal areas. And we're also looking at the ventral occipital fasciculus, which connects between the dorsal and ventral stream as well. Very, very quickly, very high variability, I should say, in the data. But the take-home message is this, is that when you look at all these pathways on the net, ventral pathways in terms of white matter volume seem to be less in CVI, same for dorsal pathways, same for this IFOF. But interestingly, the VOF, this inter-pathway communication, seems to be largely intact, which I think is intriguing because that could also be a marker. So it's not so much in terms of how your dorsal and ventral stream are developed, it's whether or not the two are communicating effectively. So finally, I just want to say thank you to a number of individuals you might imagine who are involved with this. I'm very proud to say that many of these postdocs and fellows in the lab have gone on to develop their own research, independent research programs in CVI. Our collaborators, not only nationally, but internationally, support from the Perkins School for the Blind, Boston Children's Hospital, as well as our collaborators in Italy, National Eye Institute, and various private foundations. The last thing I want to say is hopefully a call to action. We sort of had this kitchen, sink approach where we're just trying everything to try to get an idea and understanding what's going on, simply because at that time, we didn't even know what we didn't know. So we tried everything. My hope is that neuroscientists, vision scientists who are a lot smarter than I am, like all of you, will get involved with CVI to bring your expertise, to bring your thoughts to this population, because I fundamentally believe that they deserve it. Thank you.
Thank you so much, Lotfi. Great talk as always. 15 years ago and all during.
I'll see you in 15 years.
During that time. Yes, thank you. So time for a few questions. Heidi?
Thank you. It was very interesting talk.
Thank you.
So I have a question. So I understand that it's not very clear the whole phenomenon.
Yeah.
How did you diagnose?
Yeah.
And second question, have you ever tested prefrontal cortex? I just thinking like if the problem is not the information that you're getting, because apparently they don't have any problem with that, or not the big problem. And thinking that the problem is actually giving relevance to the distractors, targets, and so on.
Yeah.
Whether it could be actually more central.
Yeah.
Prefrontal cortex.
Absolutely. So great question. There's actually two questions there. The first is the diagnosis, and that is a big challenge in the community because there is no universal algorithm, if you will, to diagnose CVI. Now, with the National Eye Institute, one of the sort of ideas that we're having is, can we come together and make a consensus idea of what it means to diagnose CVI? What would you do in order to do that? I can tell you that personally, for me, it's the medical history that tells me the most. When I have a colleague or a family that says, does my child have CVI? The first thing I ask is, tell me about the birth. Tell me about the pregnancy. Were there any complications? Was there something there that raises the red flag of suspicion that there was an early hit to the brain that could explain everything that's going on. So that's the first thing. So diagnosis is something that needs to be formalized for sure. But that is sort of the grounds for the level of how we do it. In terms of your second question, I completely agree that there's more to it beyond just dorsal and ventral stream and early visual areas. I do think frontal areas become really, really crucial. This is the type of situation where the more we dig, the more the bigger, the hole, I should say, gets bigger. And I do think that frontal areas become very, very interesting in this. Just to, again, dovetail onto that point, we know that in elderly populations, the frontal cortex tends to become back online, right, with dual sensory loss, for example, almost as if all the things that used to be automatic in early visual areas, they now have to be more attentive of. They have to work harder to process sensory information that was essentially automatic before. I think that CVI is very, very similar to that, but on the front end, on the developmental side. So things don't become automatic. They have to attend that much more. And that's why these kids are so exhausted after a day of schoolwork, for example.
Yes, Ione?
So yes, I'm going to check the astrocytes in ventral stream after.
Great talk, absolutely fabulous.
Thank you.
I'm sure their astrocytes will be fascinating, but that is not what my question is about.
Okay, I saw that coming.
Actually, my question was more about sort of hidden CVI, right?
Yeah.
You know, you have a lot of kids who are premature now, 1 month, 2 months,
Yeah.
right? Their parents are told, go home, no
Yeah.
problem.
Yeah.
How much do you think this is just undiagnosed?
I think it's a massive amount. It's like many things in medicine, it depends where you live. If you've got access to individuals who are aware, we have the resources and so on, absolutely, we'll pick it up sooner. It's also interesting looking at around the world, like, so to the situation of prematurity. Prematurity is up in the United States. This is not a problem that's going away. And like it was also mentioned before, these are complex kids who have all sorts of issues, like cerebral palsy, for example. So then the motor issues tend to be really what's sort of up front and center. The visual, the language, all those sort of things kind of fall by the wayside. So I completely agree with you. A lot of it is undiagnosed.
I was just wondering about, you know, kids who we almost barely don't consider premature, like 1 month,
Sure.
6 weeks,
Sure.
right? You
Right.
know, 1-2 days in the ICU.
Right. Right.
You know, they might have learning difficulties, but nobody would ever attribute it to vision.
Absolutely. They're there. And we have many kids born term. And still later on through genetic testing or discovery of neurometabolic disorders and things like that, we then see that association. But the reality is that many of these kids fall through the cracks. And that's especially if they have good visual acuity, visual field. So they go and see their eye doctor, for example, and the eye doctor is like, okay, read the chart. Okay, TZV-ECL 2020. Eye looks good, nerve head looks good, go home. Or you know what, maybe your child has psychiatric issues. Maybe you have psychiatric issues. And now that child goes on a completely.
How did you know?
No, I wasn't pointing to you. Sorry, I wasn't pointing to you. And the horror stories that we've heard from families who've gone to 5, 6, 7 eye doctors who said your child has psychiatric issues simply because all they looked at the eye and they didn't think about the brain piece that was driven forward.
Here comes your work and the work in CVI, raising awareness about this condition.
Yeah.
And also, like Mriganka was mentioning that the only mechanism-based treatment for neurodevelopmental disorder is for Rett syndrome.
Right.
And so this is neurodevelopmental disorder, which seems to be much more broadly.
Yes.
Yeah, and this kind of work is necessary of neuroscience, because not only clinician and optician and so on, but to look at the mechanisms, which you promise. And close to Ione's question, don't be stressed. Yeah, it's not only that there is not enough awareness, but there is huge diversity, and which I think is a big challenge. So how do you approach this?
Yeah, I mean, that's a fair question. I can tell you that every time we publish or even submit grants, like that's one of the first questions that come to us. So they say, you know what, this is not even studyable. I know I'm inventing a term here, but they said this is just far too heterogeneous. There's too much going on. But the clinician in me and the public health, background in me says, that's not a reason not to do this. This is a reason to go deeper into this. And I can't possibly go to these families and say, sorry, we don't study CVI because it's too heterogeneous, or limit, for example, the profile of individuals that we work with and say, sorry, your child doesn't have the right form of CVI. We have to get in this. And I think the idea is to go in, and as we study more, as we characterize this, then maybe we start parsing the various types of CVI, maybe by etiology, maybe by level of function, and we'll have a better understanding of how there is a relationship between the cause, the etiology, and the profile as well. And then I think things will start becoming much more clear. But it's not a reason not to do it, it's a reason to do it.
Okay. Lotfi, thank you so much, and keep the rest of your questions for the general discussion. And now